After performing all the pre-processing steps and filtration and clustering and quality control, now we will label the cluster. But a question is, how will we label the cluster, and is the process same to that of single cell RNA-seq data analysis? The answer is no. And single cell RNA-seq data analysis, we first of all perform the differential gene expression analysis, and after performing the differential gene expression analysis, we have some known cell type specific markers, and based on these cell type specific markers we label the cluster. But in single cell ATAC-seq data analysis, the problem is we cannot directly look for the gene expression analysis, so the process of labeling the cluster is different, but the goal is same. So let's get started with labeling the cluster. So first of all, we will load the Serath object that we have stored in our previous session. So first of all, load the Serath object. We will use a PBMC, the read RDS function, to load the Serath object. And run. The Serath object has been loaded. After loading the Serath object, now we will create an activity matrix for a gene. So we will create an activity matrix for gene. So we will use the given line of code. And we will use the gene activity function from the SIGINAC. So PBMC. Before running the code, first of all, let me tell you that the gene activity is a SIGINAC function that estimates gene level activity scores for each cell by counting the TN5 restriction enzyme cutting sites. So now we will run the code. It will take some time. The gene, after taking some time, the gene activities or scores has been calculated, and you can see it takes almost 12 minutes. So now we will use the gene activity scores, and we will convert this into a Serath assay. So create a Serath assay, and we will name this Serath assay as predicted RNA. We will use a create assay object function from Serath to create a Serath assay. And counts gene activities and run. Now we will add the predicted RNA assay to the Serath object. So add predicted RNA assay to the Serath object, which is a PBMC. So RNA and run. Now I will show you the assay, and just click on the PBMC and click on the assay. So you can see the assay, there is only one assay, and that is the PICS for single cell ATAC-seq dataset. Now we will normalize the data. So normalize the data. So we will use the given line of code for normalization. So PBMC, we will use the normalize data function from Serath to normalize the data. So PBMC, which is our Serath object, and assay is RNA. Normalization method will be log normalize. And then we will access the count RNA. And run. The normalization has been started. This line applies a log normalization method to the gene activity assay. So the normalization has been completed. Now as I showed you earlier that our default assay was PICS. You can see here PICS, but now we will change the default assay to RNA. So first of all, I will show you the default assay by the given line of code. So we will use the default assay function PBMC and run. You can just look at the output PICS. It means that the default assay is PICS, but now we will convert this to RNA. So we will use the default assay function again and PBMC and run. And now the default assay has been changed to RNA. Now our object is ready for clustering the labels. So I will copy some codes for a cell type, in which a cell type specific markers are given. So to check the expression of those markers, so paste. Each line of code will create a feature plot. So for these types of cells, I think this may be the monocytes. So for these types of cells, we will use these cell specific markers, including the CD14, LYZ, and these, and this parameter, the max cut parameter, will keeps the color scale at the 95 percentile of expression value. So we will first of all run this line of code to check the expression of cell type specific markers to know that which cells are monocytes. So run. These will create a feature plot. Now let's zoom in. You can see the expression of cell type specific markers and different clusters, and based on the expression of these markers you can label the cluster, that which clusters are monocytes. And now we will check for other clusters. So now we will run this line of code for B and NK cells. Run. Zoom in. So based on the expression values of these cell type specific markers, you can label your respective clusters. So next we will run these line of code for the T cells. Zoom in. So you can check again that which clusters are rich and the cell type specific markers for the T cells. So based on the expression values of these markers, you can label the clusters. In our next tutorial, we will look into how to integrate the single cell RNA-seq dataset with the single cell ATAC-seq dataset.